Hello, everyone, and welcome to week 4. Now, basically, as we just saw, we're now gonna go into more of the immune system, but we're gonna basically be dissecting the immune system because, as we said, the immune system is this huge kind of, let's say, system that is not just consisting of one organ, but it's a series of cells that are working so well together in a very intricate manner to kind of, let's say, protect us from cancer, from infections, and you name it. And today we're going to be talking about two very important parts of the immune system, which are basically your physical barriers and the complement system. So, as we were basically had, this is a quite recap of what we were saying before. Between our immune system, we can basically kind of categorize things between the innate immune system and the adaptive. And the innate is much more, let's say, the first line of defense. It's fast and non-specific, meaning this is literally the first thing that's going to protect us once we are invaded, be by it from a virus, a bacteria, a fungi, or by a cancer cell. They're the ones that are able to recognize they're fast, they're there, and non-specific. And think about them. Seriously, think about, let's imagine you have your body, which let's say is a whole building, okay? You want to protect that building. So the literally your innate immune system is literally the cameras, the basically kind of, let's say, the barbed wire around the walls, these things that are literally there. You've basically they're non-specific, they're fast, they're just there as the first line of defense. The adaptive would be then basically the security guards. These are basically things that we've taught. We're like, okay, this is someone that's an invader, and they're there, and then they take a bit longer. This takes, what's it called, can take days or weeks. And of course, there has a memory. There's a memory that occurs. When it comes to the innate, it doesn't. So really, we're going to focus today a lot on the innate immune system, the chemical barriers in specific, the physical and chemical barriers, and also about the complement system, which is part of the innate immune system quite good. So when it comes to basically, let's say, the barriers, let's say from the innate immune system that we have, you can really kind of divide them into two. The physical barriers, these are like your skin, the mucous membranes, the cilia, the tears, the epithelial tight junctions, shedding of the skin cells. These are your physical barriers that protect us. And then you have the chemical barriers. These are much more the things that make, more chemicals that we secrete that are there protecting us in a non-specific manner and quite fast. These are acidic environments, enzymes, itomicrobial peptides, sebum, and sweat. We'll be literally talking about more details into each one of them. So if we look now talk about generally the physical and mechanical barrier. So if we look at the physical mechanical barrier, all of our outer and inner surfaces of the human body are literally your most important and crucial physical and mechanical barrier. This is your skin. This is the mucus, basically the epithelial kind of that basically has. So basically you have the skin, that's what I was saying. That already is the most important barrier that exists. Believe it or not, it's the barrier that actually stops most of all kind of infections. Because think about it, like we are bombarded literally uh, thousands or even millions of different types of microorganisms basically in a whole day. The thing that doesn't allow them to get inside is our skin. Our skin is the first defense, and actually majority of our innate and adaptive immune system doesn't even actually see the big portion of infections or basically microorganisms that are basically trying to attack us because our skin doesn't allow it. So it's one of the most important and crucial physical barriers that there is. But then you also have the mucosal epithelia. These are basically the things that we see around basically our trachea, our digestive systems, more of our literally kind of urinary tracts. These are also extremely important, and we will be talking about them a bit later. But we also have actions, things that move. These can be cilia. These, for example, are the nose hairs. Those are called cilia that move back and forth. And what they do is they literally push all of the bacteria, all the viruses, everything that's there outside. So it kind of prevents them from going inside. But in our bowels, there's something called peristalsis. And peristalsis is the movement. This does not allow any, let's say, microorganism from finding a place to actually sit there and colonize. Because no, it keeps moving it outside, it pushes it more and more and more. So this is why how bowel peristalsis is so important. And then you have secretions. This could be sweat. tears, saliva, mucus that are literally flushing microorganisms away or trapping them and basically kind of removing them from our system. So these are more the physical mechanical barriers that exist. And I want to really talk about the skin, because in the skin, as we know, the skin, you have a lot of these kind of, let's say, different, let's say, layers. You have the hypodermis, the dermis, and the epidermis. And between these cells that are all making up of your skin, they're so tightly kind of connected by tight adherence, this is mobile or gap junctions, that kind of creates a very common physical barrier, doesn't allow anything to go inside. It really doesn't allow it. So it's this kind of tight junctions, this adherence, this mobile gap junctions that are formed between the skin cells that doesn't allow anything just to go inside to your basically into your veins or into your arteries or anything of that sort. But within also there you have basically the stratum corneum, which is basically the outermost part of your skin. What happens there is that your Langerhans cells actually start to differentiate into these keratinocytes. So here it's written keratinocytes, keratinocytes actually start creating two corneocytes. So you have the keratinocytes that start creating basically the corneocytes. And you have these different layers of actually keratinocytes. And in specifically, you have this dead layer. This is the first layer that's on the top. And this is actually extremely important. This layer, the stratum corneum layer, this is the outermost part of the skin that are made of dead keratinocytes. These, all of them, are actually super important in protecting us. Because think about it. How can a bacteria or a virus infect dead cells? Especially viruses. Viruses need live cells to actually to replicate. Well, bacteria also in some sort of way need basically a live cells to actually do something. And think about this in a way. Can you plant actually a plant in concrete? No. Well, the same thing goes here. So the stratum corneum is extremely important when it comes to actually, let's say, preventing anything from, let's say, making a home. It also withstands a lot of mechanical forces. It prevents dehydration and it stabilizes the pH, the skin pH. So it actually gives a very strong pH there that really helps give this acid mantle that kind of prevents any, let's say, kind of bacteria to grow. Then you have the mucosa. The mucosa, as we said, these are the things that are found literally in our, let's say, respiratory kind of system, in our digestive system, more in our reproductive system, in the urinary tracts. And these are extremely important because unlike the skin, the mucosa has to allow some gas exchange, some exchange of liquids. There needs to have, because that's how we basically are able to take in liquids, to breathe. There has to happen. So because of that, there's a lot of things that are literally, moving in with the air that are able to actually cause disease. Viruses, bacteria, fungi, parasites, you name it. So there has to be some sort of protection. And the mucosa has a lot of it. So you have actually, for example, cilia. So these little hairs that are moving back and forth, pushing out anything that shouldn't be there. We see this basically actually in our intestines. We see this also in our basically noses. What you also see is goblet cells releasing this mucus layer. This mucus layer is very sticky. It really prevents any bacteria or viruses to go inside. It kind of traps them. But also you have a lot of antibodies being pushed in this mucus layer. These antibodies can bind to different types of bacteria because you just don't have one type of antibody. You have hundreds or thousands of different types of antibodies, all specific to different type of microorganisms. But also you basically have a certain pH, certain basically other antimicrobial substances that we will be talking a lot about it. Now let's talk about the chemical barriers. Chemical barriers are all these molecules that I was just talking about here. For example, basically in the mucus, that kind of inhibit bacteria or other microorganisms from actually killing or growing. And these can be, for example, acidic secretions. These can be antimicrobial peptides or lipids, proteases or protease inhibitors or enzymes. So things that can actually disrupt bacteria or viruses or other microorganisms from doing its job. So they really have this direct attack on pathogens. They can really kill them, or they can create a hostile environment for them not to be able to live. So they can have many different, let's say, mechanisms of actions. Either they directly kill, they or even actually have basically a role to enhance your immune system to kill them, or they can actually create a hostile environment for them not to actually be able to live. So the first, let's say, chemical barrier that it comes, I would say, to mind is acids. Acids are so fucking important. They literally are found in your stomach, your skin, and your vagina. And they really help you denature microbial proteins and enzymes. They help disrupt the whole membranes. They actually create, gonna alter the ionization state, and they can actually also enhance the activity of other chemicals like antimicrobial peptides or lipids to even do a better job. And really think about this. And I'll give you here a good example on how important acids can be. In a lot of women, what happens specifically in their vaginal kind of environment, it has to be acidic. Sometimes what happens in certain diseases or certain things, certain, let's say, disruptions, the acidity can go down, or sorry, can actually increase, can go down, yes, and become more basic. And this is extremely dangerous because these then women have a huge problems with infections in their basically vagina or their uterine tracts, and specifically they have a huge susceptibility to candida, which is basically a some sort of basically microorganism. It's kind of a yeast infection that really can persist and do so much harm for long periods. And this is all because of the acid. And this is just to show that how important the acid can be. But you also see this a lot with people that actually have burnt skin, because you disrupt the acid mantle so much that you're constantly having infections. So these people have to be literally put in clean rooms or environments that are, let's say, quite clean, because they have such a huge susceptibility to infections. This is how important this acid mantle can be. And then you have other stuff that are being released, and these can be antimicrobial peptides. And antimicrobial peptides, it's, let's say, it's a term, it's a big name, let's say it's in some sort of way a theme of multiple types of let's say molecules that have many different types of properties. And we basically, because there's so many different types of them, we can actually classify them, as depending on the source where they're coming from plants, mammals, or insects, on their activity. Are they anti-cancerous? Are they basically antiviral, antifungal, antibacterial? They can basically classify them on how rich certain amino acids they have, like lysine, tryptophan, or histidine, and their structure. Are they alpha helical, beta sheets, or both, and etc. And of course, they can actually have very different types of actions. They can actually kill a pathogen directly, or they can actually also modulate the immune response. They can actually cause the immune system to start kind of acting in a manner that to kill the microorganism being affected. And these are become a very, let's say, alternative to antibiotics, because as we know, or as if you guys maybe have heard, that we're having a huge issue when it comes to antibiotics resistance. That literally the last class of antibiotics, we started to see some sort of bacteria that are not being able to respond. And that's extremely dangerous, because then we have no drugs able to help us to combat the bacteria. So a lot of people are actually now investing a lot of time to finding a lot of actually antimicrobial peptides. And this is actually something really big that LAU does, and AUB. They really have a huge, let's say, resources to study different, let's say, antibacterial peptides that are coming out, either from insects, from plants, from nature, you name it. And as I said, antibacterial peptides, they can be so many different types. They can literally be defenses like there's cathelicidins. They can be basically certain peptides that are able to disrupt membranes of bacteria or funguses. They can actually be chemotatic agents. So they can actually be peptides that are able to attract the immune cell to the site of infection. They can actually induce angiogenesis. This is literally creating literally a blood supply of veins or capillaries that enhances the trafficking of immune cells to the site of infection. This can be wound healing. This, for example, let's say this is very important, where sometimes the bacteria are literally trying to destroy parts in your skin. So you have certain antimicrobial peptides that are able to actually cause the whole skin to come back to kind of create this wound healing and further protect us later on. These antimicrobial peptides can literally bind to certain bacteria or viruses and can opsonize them. And opsonizing, it literally means it's to flag it for other immune cells to come and kill it. And these could stimulate also cytokines, chemokines, or histamines, other molecules from immune cells to further combat them. And we'll be talking a lot about cytokines and chemokines a bit later. Another basically antimicrobial peptide that we see a lot this is more of a kind of an enzyme, yes, that you see actually more in your sweat and your tears as lysozymes. These are extremely important in actually removing this beta-1,4 glycosidic bonds, and these are mostly found a lot in gram-positive bacteria and also biofilms. So you're able to actually combat bacteria or biofilms to remove certain bacterial infections. And they can also, by doing that, once you remove this kind of, let's say, this peptidoglycan found in the gram-positive bacteria, you're enhancing the immune system to actually kind of phagocytose them. So it's quite important. Another thing that we also have in our tears and sweat is also lactoferrin. Lactoferrin is a antimicrobial peptide that's able to actually sequester iron. And iron is really, really important for kind of bacteria to grow. So if you sequester lactoferrin, you can actually kind of inhibit them from kind of replicating. So in generally, these antibacterial peptides, they, when it comes to, let's say, bacteria, they can have either, we call them bacteriostatic or bactericidal effects. And bacteriostatic, what it means, it kind of prevents them from multiplying. So literally, they're kind of stopping them from becoming much more. Or they can be bactericidal, the meaning that they literally kind of kill them completely. Another very interesting, let's say, kind of chemical barrier that we have are bacteria itself. Because think about this. This is kind of maybe might be very new for people here. A human body contains about 10 to the power 13 cells, but we actually harbor literally 1 log more bacteria. We have 10 to the power 14th, and that what we call is the normal microbial flora. So these literally bacteria can live in our skin, in our intestine, in basically our urinary tract. They can live literally everywhere. And believe it or not, this bacteria that are there can also be, in some sort of way, some sort of line of defense. So let me show you basically, this is kind of all the different areas where we have different types of basically flora or literally populations of bacteria. You can have them in your stomach, you can have them basically in your eyes, basically in your auditory canal, in our skin, in our foot, you name it. And again, the amount of bacteria that we have surpasses the amount of cells that we also harbor. It's crazy. And technically, they can protect us a lot against pathogens. Because if you think about it, they can actually release a lot of certain products. These can be fermentation products or acetic or butyric acids that can inhibit other bacteria from growing. So there can be certain bacteria inside our stomach that are releasing chemicals that bacteria that can be dangerous for us that prevents them to actually survive. So they can really help us in that sense. Because if you think about it, in our bacteria, there's a certain amount of space that we have. And of course, our noble flora, our noble bacteria that are there, are already occupying space there. And they don't want to let any other bacteria from the outside come in, because then they'll be competing for space. So they're literally kind of stopping them from doing anything. And how they're able to stop it is by sometimes literally releasing these bacterial products, by increasing pH, by sequestering nutrients or oxygen or body secretions. They're literally, in some sort of way, kind of competing. They really are competing with other bacteria in a sense of way. So sometimes you have healthy, let's say they're called normal flora bacteria. These are ones that are there that don't do anything. They're really actually helping us to live. And those guys are trying to compete with the opportunistic bacteria, these other bacteria that are literally coming from the outside from doing anything. So they're trying to help us actually in some sort of way. And in humans, at birth, or sorry, basically when we are at the womb of our mother, we actually don't have, we are sterile. We don't have any bacteria with us. It's only when we are actually, are born that we actually get a microbial flora. We're infected with all of these different types of bacteria that are literally then going to colonize every single part of our body. And there was a very interesting study because, of course, people can be born through either the C-section, meaning that there's an incision in the stomach that you remove the baby through there, or basically others are more born through the vagina. Basically, you're delivered through the vagina. And of course, depending if you're actually born through the vagina or you're born through the stomach, you get colonized with different bacteria, because if you look at here, the bacteria in our stomach is very different to the ones that we have, for example, in our vagina. You get different populations. And this can actually have many different effects. So they saw that if you actually are born through the vaginal route, you have a much more diverse, much more healthier bacteria that are colonizing everywhere. But if you're born through a C-section, you have bacteria that are coming from the stomach that are not actually the best. They're very, let's say, limited. There's not a huge diversity, and this actually can have a lot of effects during how you grow. These, for example, people have an 80% chance of actually getting asthma. So they also have huge other problems when it comes to, for example, allergies or autoinflammatory diseases. Because literally this kind of bacteria is in some sort of way really helping our immune system a lot to kind of educate it. So it's important sometimes the bacteria that we are basically developed with, and it all depends on how we're born. Like, are we born through the C-section? Are we born literally through the vaginal route? It depends, because different routes have different bacteria that you collect then at birth. So as I said, the effects of this normal flora, it prevents the attachment and basically penetration of microorganisms, so they really don't let them. They can also produce antibiotics or certain antimicrobial peptides that doesn't allow actually any other bacteria to come in. It also helps actually the immune system to kind of push it to kill, to like, hey, we have an infectious bacteria coming in. It also produces a lot of enzymes and vitamins that can help our immune system per se, or have a direct effect in killing the bacteria, and also can help us in our own metabolism. But sometimes the flora can be also nasty, because what happens is that we can also harbor in our flora certain bacteria that in normal circumstances they don't do anything. They literally are harmless. But in certain circumstances, they can actually become opportunistic infections, meaning that they're waiting for a certain opportunity to become pathogenic. And that's an example of E. coli. So what happens sometimes is that in a case where we have a disruption in our urinary tracts, meaning there's a lower amount of pH, there's some, let's say, defense systems that are gone down, the E. coli senses it, and now suddenly it can move from your stomach and go there and cause a lot of infections. So sometimes it's not always the good things that come out from our flora. Sometimes they can be bad, depending on certain opportunities that it actually there is. So now I've spoken a lot about the mechanical barriers, physical barriers, and also the chemical barriers that exist. And these are the first thing that protects us from literally kind of infections. Now let's talk about the complement. The complement is a very important part of the immune system. Quite of a, how can I put it? Quite complex, but my favorite in my opinion. So what is complement? Well, complement is actually, this is literally coming straight from Wikipedia. It's known as literally a system that has literally, so I'm sorry, I'm going to just read this. It's the complement system, also known as the complement cascade, is a part of the humoral innate immune system that enhances or complements the ability of antibodies and phagocytic cells to clear microbes and damage cells from an organism, promotes inflammation, and attack pathogen cell membrane. So the complement, literally, to make this more simple, is a series of 20 proteins, 20 different proteins, that what they do once they're activated, they can actually increase our own immune system to phagocytose or clear certain microbes, can promote inflammation, and literally attack directly the membrane of certain pathogens. That's what it is. It's a series of proteins that form this cascade that they start cutting each other and they start to actually create three actions, either causing phagocytosis, causing inflammation, or actually directly killing the pathogen cell membrane. So, as I said, they're a collection of circulatory proteins. They're literally all around their blood, specifically in our serum. They're part of the innate immune system, and they literally, as their name says, they kind of complement the adaptive immune response because, as I said, they really help macrophages or antibodies to do their work really well. So they complement the adaptive immune response, specifically the antibodies. But they also can actually create this beautiful pore that's seen over here to literally attack the cell membrane. But they can also kind of create an inflammation, kind of call other immune cells to come to the site of infection. And we'll be talking in much details about the complement. So when it comes to the complement system itself, it can be activated through three different pathways: the classical, the lectin, and the alternative pathway. But all three pathways involves C3. So every pathway starts differently, but they all end up here at C3. And what they're trying to do is they try to create some sort of enzyme that's able to cleave C3 into C3A and C3B. And I'm going to give you examples on how each pathway starts off with, okay? So let's start with the classical pathway. The classical pathway, it always starts off with an antibody or basically a C-reactive protein that is bound to a microbe. So in this case, it's an antibody. So this antibody, either it's IgG or IgM. It cannot be IgA, it cannot be IgE, it cannot be IgD. It's either IgG or IgM. So once these antibodies are bound there, these antibodies, IgG and IgM, have something called a C1q binding site. IgA and IgE and IgD don't have that. It's only IgG and IgM that have a C1q binding site. And as the name says, it literally it provides a binding site to C1q, to the C1 complex. So C1, what happens? Literally, the C1q binds to the antibodies, and once the C1q literally is bound, it gets activated, and basically it kind of activates C1R. And C1R becomes activated, and it cleaves C1s. And once C1s is then activated, C1s is able to cleave C4 and C2. So what happens is basically C4 gets actually cut into C4B and C4A. C2 gets cut into C2B and C2A. So again, I'll repeat this again. You have a C1 complex, and C1 complex is literally made of a binding protein called C1q and two inactive proteases, C1R and C1s. They're inactive unless the C1q basically gets activated and then basically kind of activates these guys over here. So the C1 will bind to the antibodies that are found in the microbe through the C1q because it's a binding protein. Once that is basically bound, it gets activated and it binds to, it kind of activates C1R. C1R gets activated and then it kind of activates also C1s. Then C1s is now able to basically be activated, and C1s is then able to cleave or kind of, let's say, cut C4 and C2 into C4A and C4B and C2B and C2A. Okay, great. But what happens is that C4B and C2A can now bind to each other, and they create something called an enzyme called C3 convertase. So this is then the enzyme that is then able to cut C3. So as we all said, it all ends basically with C3, where C3 has to be cut. So that's how the classical pathway starts. It basically, you have a C1 complex that binds, which then is able to create a C3 convertase to cut C3. The lectin pathway is very similar, But the lectin pathway uses a different complex. It doesn't use a C1 complex. It's called actually this MBL complex. And MBL is basically a mannose binding lectin. That's what it stands for. And literally mannose binding, it literally binds to mannose. And mannose is a sugar that's extremely rich in certain bacteria. So what happens is that this mannose binding lectin comes and it literally binds to basically these kind of mannoses, and they basically this mannose binding lectin activates two proteins or two basically proteins or complexes that it has, this MASP1 and MASP2. And similar to what we saw with the C1 basically S, it cuts C4 and C2. So you have C4b, C4a, C2, which is cut into C2a and C2b. So C4b and C2a come and create C3 convertase. So literally the lectin pathway and the classical pathway are extremely similar. That what they do is once they get activated, once they bind to something in the microbe, the classical has to bind to the IgG antibodies, to C1, or basically the lectin, the MBL has to bind to the mannose that's found in the microbe. They get activated and they cut C4 and C2 to create the C3 convertase. So that's where they converge with the classical pathway. So they have a different start or different recognition and different basically binding proteins, but they all basically end up in creating the C3 convertase. Then you have the alternative pathway. This one is quite a very interesting pathway in my opinion, because what happens is that C3 by itself spontaneously, there's some sort of hydrolysis that happens. So meaning hydrolysis is basically water that's removed. So this is basically kind of cleaved, water is removed, and you have C3b. But what also happens is that factor B is basically there's a factor B in our blood that it also gets cleaved by factor D. So factor D cleaves factor B, and then basically that is then it gives you BBB, which is then able to actually bind to C3b. So the binding of C3b and the cleavage factor B gives you basically the C3 convertase. So again, let's say kind of put this in a more, let's say, understandable manner. You have two different ways that's happening. You have basically the spontaneous hydrolysis of C3 into C3b and C3a. Factor B is then cleaved by factor D, and then factor B, one of the parts that's cleaved is called BB. So C3b and the BB come together and they create C3 convertase. And this is where they converge all together. So they all have very different ways of starting. The classical pathway starts with the C1 binding to the antibodies, that's then cleave C4 and C2 to give you the C3 convertase. The lectin pathway starts off differently, where you have a mannose binding lectin, but you have a very similar convergence where they're literally cutting C4 and C2. So lectin and classical pathway, their C3 convertase is the same. It's the C4b2a complex. Alternative, no. It's actually the spontaneous hydrolysis of C3 and the cutting of factor B. Once you cut factor B, you actually have the BB that's binding to C3b, and that's the C3 convertase. So it's different, it's distinct. So the lectin pathway and the classical pathway have a similar C3 convertase, but it's different to the alternative pathway. Okay? So this is how you start, literally, when it comes to the complement. This is how they can get activated, either through antibodies, either through the mannose, or basically spontaneously, which then you create a C3 convertase that is cutting C3 into C3a and C3b. So as we said, now you have the C3 convertase, and as we said, basically the classical and the lectin, they have exactly the same. You have the C4b and 2a, the C4b and 2a, exactly the same, but this one you have a different one. And this per se, basically the alternative is C3b and the BB coming from factor B. And what they do, as their name is, the C3 convertase, it cuts C3 into C3a and C3b. That's what it does. So as we said, the C3 convertase cleaves C3 into smaller subunits, C3a and C3b. C3a is a chemotaxis and inflammation agent. So C3a, once it gets released, it's literally a chemotaxis and inflammation agent. It creates inflammation, but it also brings other immune cells to the site of infection. That's what basically C3a does. C3b can basically bind to the pathogen and can actually be used as an opsonization agent. So it literally can bind to a microbe and kind of flag it so other immune cells can come in and actually kill it, either through NKs releasing granzymes or, for example, macrophages to come in and actually do phagocytosis. But it can also basically, the C3b can be part of a new C3 convertase and actually help to create a C5 convertase. So in the case, basically, as we said here, C3a is cut. C3a is needed to actually bring basically immune cells, so chemotaxis, and create inflammation. C3b can actually be used to actually opsonize. It can opsonize certain, literally, kind of, let's say, this kind of bacteria or other pathogens, and actually help phagocytosis, or basically NK cells to kill. But the C3b can also then come here and create something called a C5 convertase, which can then be used to do a membrane attack complex, which we will dive into it there. So, when it comes to C3, C3b can basically then go and bind to the C3 convertase. So basically now suddenly you have C4b, 2a, and then you have a C3b that's coming in and binding there. And you create something called a C5 convertase. So as the name says, it literally is cutting C5 into C5b and C5a. C5a, similar to C3a, it's also a chemotaxis agent. So it literally brings immune cells. But C5b is able to bind to C6, C7, and C8, and also C9, and where they create something called a membrane attack complex, which we're going to talk a bit more. So C5b, as we said, so as C5 gets cut by the C5 convertase complex, it gets to get cut to C5a. C5a, as we said, is a chemotaxis agent. It literally brings other immune cells. C5b literally comes and it literally binds to C6. Then this C6 binds to another C7, which then binds to another C8. So this then basically is able then to insert itself into the membrane of a bacteria. And it's able to do this through the hydrophobic regions of the C8 and also the C7. So C7 and C8 have hydrophobic regions that are able to bind to the phospholipid membrane over here. So that's how usually this can do. So that's why if you see the C6 and C5, they're either in the phospholipid membrane or in the phospholipids, in the phosphate heads, or outside, because it's only C7 and C8 that are able to bind to the lipid parts. And this then allows C9 and basically 10 to 16 of them to come in and create this beautiful pore. So I'm just seeing if there's actually a nice picture of this here. So as we see, the C5 and C6 are the outside. C7 and C8 are actually really penetrated inside, as you can see here from the yellow and basically the green over here. And they allow these C9s to come in and create this beautiful pore. And this pore, what it's doing is literally allowing all the water to get inside. And what happens is that the bacteria cell actually can pop. It literally pops completely. So this is what happens, basically, with the membrane attack complex. So as we said, the complement results in three things. In the membrane attack basically complex, which only happens with gram-negative bacteria and enveloped viruses. These are the only ones. They can also actually happen in cancer cells, but when it comes to infection, they can only actually, the membrane attack complex will only actually come out in gram-negative bacteria or enveloped viruses. They also help in opsonization. As we said, there's C3b, and C3b can bind to bacteria. And what that does, it actually allows macrophages to come and actually kind of, let's say, phagocytose them. Because now the macrophages are able to actually be like, we're able now to eat them out. And as we said, the C4a, the C3a, the C5a, these A's that are coming out, they literally promote inflammation. So this is the nice thing of them. They can promote inflammation and chemotaxis. So when it comes again to, you have these, as we say, the proteins, these like C1 or C2 or C3, C4, these ones that we were talking about, they're basically large subunits which are bound by something. So once you cut them, you have CXB or CXA. So X denotes the number. This could be 3 or 4 or 5, as we were saying. So usually the large subunit binds to something. So if you look basically at everything here, you see basically the B's are literally always binding. So the C4B, the C3B, the C5B, or basically they're always binding to something. The only exemption is the 2A. The C2A is the only exception that is able to bind. But the CXA, these are the smaller subunits that are broken. These are more cytokine and chemotaxis kind of ones. So these are, for example, the C4A, the C3A, and C5A. Again, the exception is C2. The C2A basically is able to bind. So this is just to help you understand or to memorize that, okay, the B's, so when you basically get the C proteins, once they're cut, the B ones are the ones that are literally binding together to create these convertases. The A's are more the ones that are needed for the chemotaxis or the subunits, or the cytokines. So as we said, the C3A, C5A, and C5A, they literally are inflammatory response mediators. They can actually cause contraction of blood vessels, increase permeability, cause degranulation of mast cells. So they're all creating an inflammatory response. They literally are bringing, basically, when you contract the blood vessels, you're pushing blood flow. You're also making the blood vessels permeable. So basically what happens is that, let's imagine you have a skin infection. You're causing the immune cells that are literally in your blood vessels to be more permeable. So they're able to actually, travel from the blood vessel into the skin to where the bacteria is. It also helps mast cells to release histamine and heparin secretions that are also important to combat bacteria. The C3A and C5A also are able to bind to more specifically C3A is able to bind to bacteria to help phagocytosis, but C3A and C5A can also bring other immune cells, which is basically called chemotaxis. So this is literally the beautiful sum up. Let's actually, let's really kind of go over it. So as we said, you have the classical pathway, the lectin, and the alternative. The classical pathway always happens when basically you have literally the C1 complex, which is made of the C1Q, 1R, and 1S. So what happens is C1Q literally binds, which are these literally green little kind of tentacles that bind to the antibody. And what this does is they literally activate the C1R and C1S to start cutting C4 and C2. The lectin has a very similar process where you have this basically mannose binding lectin that once it's bound, it literally activates the MASP1 and 2 to cut C4A and C2B. So it's cutting C4 and C2, and then you get C4A and C2B being released. But importantly is that C4B and 2A are literally coming together to create the C3 convertase. When it comes to the alternative pathway is a bit different in where this has spontaneous. So there's no binding to anything. The C3 hydrolysis is literally happening in where hydrolysis is being removed. And what that does, basically that kind of tells the factor B to start to be cut. And the factor D does that. Factor D comes in, it cuts factor B. And what happens is that the BB coming from the factor B binds to the cut C3B that's been spontaneous being cut. And now that's literally your C3 convertase. So once you have the C3 convertase, you start to cut basically C3, and then you get the release of C3b. C3b basically comes and binds to the C3 convertase, which then literally becomes a C5 convertase, which cuts C5 into C5b and C5a. C5b is then able to bind to C6, 7, and 8, which then attach to the membrane of your target. This, in this case, can be a gram-negative bacteria. As we said, it only can go into basically gram-negative bacteria. And that allows the C9s to create this really nice, beautiful membrane attack complex. One thing that I want to also say about the alternative pathway, we used to think that it was really a whole pathway by itself. we started to believe that this is not true. We believe that it's more of a amplification loop. It's actually there to help either the lectin pathway or the classical pathway to provide more C3b. So it's an amplification loop to create more C3b, because as we know, we need more C3b for opsonization, but also to create the C5 convertase. So now we started to see that we don't think that the alternative pathway is something that only happens by itself. No, it only happens when either the lectin or classical pathway is activated and is there to further kind of supplement it with more C3b. So it's literally there as more of a kind of an amplification loop. And now there's been a lot of actually research around factor H and how that factor H is able to kind of, let's say, regulate this alternative pathway. And I didn't speak about factor H, and I don't want you guys to know much about it, but factor H kind of prevents the alternative loop from happening. But don't worry about it right now, about this. But what I want you to know is mostly that the alternative pathway, it's now the new research has been showing that it's not really, it doesn't really happen by itself. It happens together with the classical or the lectin pathway, and it's just there to support the production of more C3b. If you guys have any questions, please reach out to me either by email or WhatsApp. Thank you so much.